Ladies and gentlemen, hello and welcome to yet another informative video on HIV, specifically the life cycle of HIV. Now, drugs for HIV do not cure or treat HIV. Okay, what they do is they make sure that the virus does not multiply or over multiply, and through that they limit the production of new viruses that will actually end up attacking more of your immune cells to actually lower your immune system. So what they do is they block that multiplication of this virus so that you don't have a weakened immune system. And that is target number one for drugs for HIV, to make sure that they improve the quality of life and they improve your immune system. And then number two, they make sure that the viral load is very low, which you call the undetectable level. And when you get to that level, even if you end up having unprotected sex with somebody who is HIV negative, chances of you spreading it to them are very, very low. If any case they are not there, it's not possible that when you have a low viral load or undetectable viral load, you can transmit HIV. So therefore, these drugs were designed to improve your immune system, to improve the quality of life, and to limit the spread of HIV. So those drugs act on different stages of the life cycle of HIV. And I want you to see this, for HIV to actually destroy your immune system, because we don't die from HIV, we die from AIDS. HIV is the virus, AIDS is the complications or the group of diseases or conditions that come in as a result of the weakened immunity by the virus. So the virus, its role is just to weaken your immune system, and then through that now you are prone to attack, or you're liable to attack by other things like TB, like pneumonia, like meningitis, and even cancer. These are the things that actually sweep you off, and specifically TB, very dangerous. So now I want you to see that for this virus to actually attack more cells, it must produce more copies, and it's very smart. So it uses your cell, which is the CD4 cells, to actually create more of its viral copies that will go attacking all other immune cells in the name of CD4 cells. So I want you to sit back, enjoy this, and we'll have, of course, our own questions. Why this therapy does not cure HIV, and why or how is it possible that we can actually lower the viral load to a level that is very undetectable in system, but we can't clear HIV? So we'll get to ask those questions, and we hope that as you interact with us, you can actually help us understand this even further, because all of us have to actually make a collective thinking about this. We don't have to rely to the information that is coming out instead of actually opening our heads and thinking, because common sense applies more to this than any other thing. So this is me taking you through the HIV life cycle, and also going to take you through mode of action of these drugs, the drugs that are called the antiretrovirals, the ARVs, and then you get to understand how they work and where they work specifically. But I want you to know this before I even start it. We have several steps here in this life cycle. The first step is binding and fusion. The second step is reverse transcription, and I will tell you what happens throughout these processes. The third step is integration. The fourth step is transcription, and then assembly of the viral protein and materials, and then now budding off to go and release the new virus. But this virus is coming in with multiple enzymes, 3 enzymes to be specific. One is what we call the reverse transcriptase enzyme. The second one, we call it the integrase enzyme. And the third one is called the protease enzyme. So once you understand those enzymes, you will know their roles in this process. So let's start. Step number one, we have a free virus. And this is the structure of a virus, just a general structure of a virus. The genetic material is encompassed in a protein shield, and that protein shield has these spikes that you see, and those spikes, we call them receptors. Now understand this, on the virus that is HIV, the receptors have a name that is called GP120. And somebody will ask, how then does this virus recognize CD4? Because this virus does not attack any other cells in the system apart from the CD4 cells. So then somebody will ask, how does the virus know that this is a CD4 cell? It doesn't have to know that. What it needs to identify is to have this GP120 receptor that is coming, or protein, sorry, that is coming in to attach to a specific receptor on the CD4 cell. That receptor is called a CD4 receptor. Now look at it this way. We have these receptors all over the surface of the CD4, because this is the CD4 cell, this is in the nucleus, and this is in the cellular matrix. So we move from the cellular matrix into the nucleus of the CD4 cell, and this is the outside part of the cell. So this cell, along this cell membrane, it has these receptors that are supposed to identify the GP120 protein. And that receptor is called the CD4 receptor, but it is actually encompassed between two other co-receptors. The blue receptor is the one that we call the CCR5 receptors. Do not cram these names. Okay, so we have a receptor or a co-receptor that is called the CCR5, and then the red one is called the CXCR4. I will tell you why I'm mentioning this, because there are drugs that actually block this so that we don't have the virus attaching itself to the cell. So the CD4 receptor is the one that receives this GP20. So therefore, if this is the GP20, the CD4 receptor must assume or must take the shape of this GP120, so that when it comes, it fits in, because it's like a key and keyhole hypothesis. So the key comes in and fits in the keyhole perfectly so that we can open the door. So this is the one that comes in to attach to this receptor. And once it attaches to that receptor, now we have access to the inside part of the cell. But remember, that is stage 1, which we called the binding and fusion. So it must bind, attach, and then it fuses with the cell membrane of the CD4 so that it can easily release its genetic material into the cellular matrix of the CD4 cell. Now, this genetic material for the virus is in RNA form. Remember, it is the one that is carrying the instructions to make the CD4 or to instruct the CD4 cell to produce more of the viral copies so that they can actually attack more CD4 cells. So it carries that instruction. So once we have bound it here, we have fused it with the cell membrane of the CD4 cell, now it's time for us to release this nucleic material into the matrix of the CD4 cell. Now this, I told you already, it's an RNA. So for you to actually make this RNA to be active so that it passes the instructions to the CD4 cell to produce more HIV copies, we must convert that into DNA. Okay, and this process is a totally opposite of the norm, because we need to actually combine DNA into RNA instead of changing RNA into DNA. So we do the opposite. That's why it's called reverse transcription. We reverse transcription. So we do the reverse. So normally we need to move from DNA to RNA, but this one we are moving from RNA to DNA to make it active. Once we've made it active, remember our first enzyme was reverse transcriptase. Do you remember that reverse transcriptase enzyme? It is the one that does this job. It is the one that actually converts the viral RNA into the viral DNA. Now this DNA is now active, and now it moves from the cellular matrix into the nucleus of the CD4. Okay, remember the nucleus is the machinery of the cell, is the engine of the cell. So now the DNA has to move into the nucleus where we have more DNA of the CD4 cell. And the reason why we are moving this DNA into the nucleus is because we understand that we need to combine this viral DNA, which is active, with the host cell, the CD4 cell DNA. So now we have the viral DNA and the host cell's DNA combined together, and now we have a long DNA of the two. And that is very important, because this is the one that will start producing more of the proteins that will give us a new virus. So DNA comes in and it's combined with the DNA of the cell, the CD4 cell, and whatever converts that, or whatever combines this is integration. So therefore the enzyme that is involved in integration is called the integrase. That is our second enzyme. So first enzyme converts RNA of the virus to DNA, and then the DNA goes into the nucleus of this CD4 cell, combines with the DNA material, because inside the nucleus is where the cell stores the DNA. So therefore we have CD4 DNA combining with viral DNA to form a long chain DNA, and this role is actually played by the viral enzyme that is called integrase. So remember, this virus was very, very sharp, because it comes in with its own enzymes, the 3 enzymes, to actually do its work. And now here we've combined the viral DNA and the cell DNA. Now we are good to go. So we have now to multiply, to pass instructions to the CD4 cell, now to multiply more proteins and produce longer chain proteins that will be used to form the new viruses. So once we've had this integration, this will be used to form new proteins, and these proteins are here. So once it's leaving the nucleus, it is leaving as longer chains of proteins that are going to be clipped into smaller chains so that they can be released as a new virus. And whatever clips this into smaller chains is the next enzyme called the protease enzyme. So first enzyme converts RNA to DNA. Second enzyme combines the DNA for the virus with the DNA of the host cell, the CD4. Third enzyme the long chain proteins that are produced after the instruction has been passed, that are coming out as long chain of proteins, have to be clipped into small chains of proteins to go ahead and make new viruses, viral copies. That is where protease comes in. So protease, from the word protein, it breaks down these protein chains to form smaller chains, and now the virus comes out as a whole a virus, a new virus. But remember, these ones have to be assembled together so that they can form a new virus, and this is the stage that we call the assembly stage. So when we are moving this combination of this DNA into the long chains, that is what we call now transcription. So we are moving DNA into another DNA and the RNA of the new virus that is coming in, and that's what we call the assembly stage. And then now the budding is when this new viral material pushes into the cell membrane of this CD4 cell to form a bud. It's like budding. Okay, so it buds out and then it's clipped out and goes out as a new virus to go and attack more CD4 cells, and the same process continues. But remember this, once it leaves the cell, this cell remains destroyed. And that's how we start lowering the CD4 count, because it conquers. Once it conquers, it's able to multiply to release new viruses, and then now it destroys this host cell. Once you destroy this, the level of CD4 starts going down, and that's how your immune system goes down. And now this one goes and affects or infects new cells, the CD4 cells, through the same mechanism, goes through the same procedure, and as it does that, we produce more of the viruses, new copies of the viruses. So once you've understood this is the life cycle of HIV. It is that simple. Do not complicate it. Now, to the questions. Question number 1, we have all these 6 stages. We have bound on the cell membrane. We have we have done reverse transcription. Now we have produced the DNA of the virus. We have integrated the DNA of the virus with the DNA of the host cell, the CD4. And then we have transcribed the DNA, the 2 DNAs that are combined together to produce more proteins that will be used to form new viruses. And now we have assembled these proteins and cut them into small proteins, assembled them into one, and made a new virus. And the virus buds off and leaves the cell, destroying the cell behind it. So once we have all these stages, so the questions are coming in now this way. 1, we have drugs designed to work on multiple stages on this life cycle. And this is where the challenge comes in, and this is how it gets interesting. Because look at it this way. At the entry or binding or fusion stage, we have 3 receptors, or 1 receptor and 2 co-receptors. We have the CD4 receptor, and guess what? There is a drug that is designed to actually block this receptor so that the virus cannot get access to this. So the drug that blocks here, let me just use a mark to show you. So there's a drug that actually blocks this. And that drug is called abalizumab. So this drug blocks here. Let me just write it down because most of you will ask, Daktari. So we do this. We have abalizumab. This is the first drug that blocks the attachment of the virus to the receptor that is called the CD4 receptor. We also have other drugs that block these smaller receptors. For example, the 𝐶XCR4 receptor is blocked by other drugs, but I will not mention those because they are under research. So these ones are still under research and they are coded. So they don't have names, they just have codes. But the CCR5 receptor is actually blocked by a drug that is called Maraviroc. Good. So now we have a drug that is blocking the virus from attaching to this CD4 receptors. So then the question comes in. We have these drugs. They block the virus from coming into attachment with this. There's also another drug that is called enfivrotide. This one is called the fusion inhibitor. So it inhibits the fusion of this into the cell membrane of this. So that is one. Let me reserve the question until I finish these drugs, okay? So this is the first 3 drugs that block the attachment of the virus to this cell, the CD4 cell, okay? Number 2, in case the virus has already accessed and fused with the cell membrane of the CD4 cell and it has gotten access into the matrix, and now the reverse transcription is happening, we have drugs, actually several groups of drugs that block this enzyme called the reverse transcriptase. That enzyme that converts the viral RNA to DNA, we have drugs that actually block that enzyme so that we don't continue the life cycle. And these drugs, they are classified into multiple groups. One, we have what we call the NRTIs. So nucleotide or nucleoside reverse transcriptase inhibitors. And then number 2, that is one. Number 2, you have the non-nucleoside reverse transcriptase inhibitors. These are two groups of drugs that are actually first-line drugs in HIV therapy, and their role is to block this enzyme so that we don't go ahead and form the active DNA of the virus. Now, these drugs on the the nucleoside reverse transcriptase inhibitors, we have multiple drugs. Actually, here we have a lot of drugs. We have the most safest drug called the lamivudine, it's called 3TC. We have zidovudine. We have another drug that was withdrawn because it used to cause this lipodystrophy, abnormal deposition of fat. So it could get fat from the buttocks and deposit on the shoulder. So people had this buffalo hump. It also drained and made their legs so hard. So you would easily see this one is on ARV therapy. So here it's called stavudine, so it was withdrawn. We have another one called zidovudine. We have another one called tenofovir, the TDF, which is basically first-line medication of this. So a lot of drugs are coming from the NRTI's classification. And then on this other one, the non-nucleoside reverse transcriptase, we have two. We have nevirapine. Actually, we have a lot. We have nevirapine, the most commonly used, and we have the EFV, efavirenz. Most of you have had an encounter. If you worked in a CCC center, possibly you've had an encounter with this medication. Or if you're somebody who is living with HIV, you must have known this medication because they combine maybe two of this plus one of this. So two NRTIs plus one NNRTI. Okay, so this is what you see. So we have now drugs to block this process. And now, in case the virus has already proceeded from this stage and it has come into the nucleus, and now we are converting the DNA, combining it with the host DNA. There's a drug, because this is an enzyme called the integrase. And now we have drugs that are called the integrase inhibitors. So they inhibit the enzyme integrase, integrase inhibitors. And this is where it gets interesting, because every step is inhibited by a drug. So in the integrase inhibitors, we have two drugs. We have dolutegravir, which is one of the, nowadays is actually used in first-line therapy, and also is used for PEP, dolutegravir, DTG. That one has complications with blood sugars. It can actually raise your blood sugar. So therefore, people who are diabetic have to be very cautious about that. They are told, I believe so. And then there's another one that is called raltegravir. So we have two here. We have raltegravir, and then we have dolutegravir. Okay, so these are the drugs that are actually stopping the integrase. So we don't now incorporate the viral DNA into the host DNA. Okay, now the enzyme, the virus has already surpassed all these stages. We are now even producing the long chains of proteins. Before we clip those long chains of proteins, we have this enzyme called the protease that clips these long chain proteins. We have drugs that actually block that enzyme. They are called protease inhibitors. So they inhibit protease, the enzyme. And these ones are also a group. We have indinavir. We have lopinavir. Lopinavir is famously known as Kaletra, and is actually given as a suspension most people, in most children. But it was also used as PEP previously. So lopinavir, but it used to have a lot of vomiting and nausea. So you will vomit your lungs out for taking this medication as a PEP. So you will never do that again. Then we have another one that's also was used as PEP. I hope it's still being used. It's called atazanavir, ATZ. So atazanavir, okay? So these are the drugs that actually block here, here, and inside the nucleus. And also after we produce the proteins, and now we want to clip them into smaller proteins to encompass them and assemble them into one to release them. So all through, up to the time we are releasing the virus, we have a drug blocking each step. The only place we don't have drugs is when we are releasing it out in terms of budding, and when we have a new virus that's going to attack other cells. So we'll simply block it from attaching to other cells. Now look at it this way. So we have these drugs blocking all these steps. But we don't have therapy for HIV. But we'll still say that HIV drugs, the antiretrovirals, are incapable of clearing the virus when we have a single drug at every step, actually multiple drugs at every step of this process. We have a drug that is blocking the binding fusion process. We have multiple drugs that are blocking the reverse transcription. We have multiple drugs that are blocking the enzyme integrase. We have multiple drugs that are blocking the enzyme that is called protease at the assembly and clipping stage. But we are unable to to cure HIV. Does that even make sense to you? That is question number one. Question number two, how is it possible that we can actually do all this and then lower the viral multiplication? We lower the production of new copies of HIV virus by blocking this process. But we are unable to treat the HIV. And how did, my question exactly, how did we end up having this chance to lower the viral load to a place that is very undetectable, but we cannot destroy the same virus? A good question, right? Because if we can lower the virus, how did we lower it to undetectable levels? Yes, we blocked this process, and then we started producing more white blood cells that actually came up. How is it possible that those white blood cells cannot overwhelm these infected white blood cells? That is one. Number two, if the white blood cells are actually coming up, our immune system is actually coming up. We know the cells in our system have a life cycle, right? They have a lifespan. Let me use that. So if they have a lifespan, they will live, they will be destroyed. So how is it possible that we can have new cells that don't have the virus because of what we've just blocked here, but the same cells will still be available? These other older cells will still be available for a lifetime. I think these are questions that we need to ask so that people who have information about this can actually bring in the answers. And now that they've stopped, or there's a challenge with the US aid that is actually being concerned with this, I think we should get some answers from you people to just explain to us how this is actually happening. And you see, we have multiple drugs. The 𝐶𝐈, and the ones that are inhibiting this budding are under research. So my question is, even those which are under research, they are not coming in to heal the HIV. They are coming in to actually slow this process down and to maintain this HIV in the system, even when these cells are supposed to be destroyed. So then how are we going to get better? How are we going to trust these processes of healing? And now, another thing I've noted is people will be told and advised on how to use this medication all through. That's a good idea. That yes, we want you to lower the viral load to improve your immune system and to stop the spread of HIV. Perfect. We've even gone ahead to give you drugs that will act as post-exposure prophylaxis after the sex and pre-exposure prophylaxis before the sexual encounter. So we've stopped here, and we've also stopped in case you've had the exposure already. But we can't treat this. We can't treat this, but we have all these processes. Anyway, that is beyond me now. So this is me telling you the life cycle and how drugs work. Basically, now you know the mode of action of this medication, and this, if you're a medical practitioner or a pharmacist, you now understand it even better. So I want to tell you this. As you're taking this medication as a HIV person, and this is not me telling you to drop medication, this is me actually opening your eyes so that you can actually ask your provider questions accordingly, and depending on how you feel about this also, because I have my doubts and I ask my doubts through this. So you can ask your doubts to your doctor using these videos. Now, so we focus so much on telling you how to take the drugs and when to take the drugs, but nobody focuses on how to boost your immune system, because for HIV we have two things. We have the virus, and then we have our immune system. The challenge is, we will tell you how to take the drugs, and all these drugs come with different side effects. They can actually mess up the liver. But we don't tell you how to boost your immune system. We don't tell you to fast, because again, we scare you. If you take this medication without eating, you will get into more problems. But we know fasting is an immune booster, so we don't tell you how to eat healthy. We tell you to eat fruits when we know fruits are going to lower your immune system. So we don't tell you to eat healthy, to drop the inflammatory foods. We don't tell you to go and exercise and how to exercise, basically the strength training. We don't tell you to fast, because anybody who eats must fast, and fasting is an immune booster. We don't tell you to actually even do simple things like walking. We don't tell you what to do to boost your immune system. But we are quick to tell you to take medications. Once you've tested positive for HIV, everybody is just guzzling about medication. Then they tell you exercise, eat fruits, and all that. But nobody is telling you the real thing. So everything that we preach in this platform, avoiding the inflammatory foods, eating the animal proteins, the fatty proteins, eating the vegetables, and using the animal fats to make your meals, dropping the processed foods. This is us trying to tell you how to boost your immune system so that this cannot overwhelm you. Because remember, once this has overwhelmed you, you are now prone to attack by anything, including a common flu. Okay, so we need you to boost your immune system as you're taking these medications, so that one, you protect yourself from the side effects of this medication, you clear them off by the liver, and number two, you boost your immune system so that any opportunistic infection that is coming in is actually blocked. So the pneumonia, the TB, the meningitis, the Kaposi sarcoma, the cancer of the skin in HIV people, the common colds, a lot of conditions that are coming in as a result of your lowered immunity, we call them opportunistic infections, can be held back by just a heavy and tight immune system. So focus on both. If you're already taking this medication for HIV, continue taking them. However, focus on improving your immune system tremendously. Go work out, take those cold showers, enjoy the sun, have good sleep, okay? Ease up on stress levels and eat healthy, and then fast. That is exactly what you need to do to understand or to actually navigate through this process easily. So again, this was a mind-opening video for you. I have my doubts. Now I've asked my doubts. Kindly in the comment section, be kind to answer us these questions so that we can actually learn from you as you learn from us. Thank you so much for watching, and we'll see you in the next.